Hi, I'm Stephanie,
And I'm Keith.
and we're students with Washington University's Young Scientist program. And today we will be going through our diffusion and membrane permeability teaching kit. The purpose of this video is to serve as a tool for you, the teacher, to try to figure out how you might want to go through this lesson plan with your students. So in the video, we'll be going through the necessary background information and the safety protocols, and then we'll actually go through each one of the demos so that you can see how we envision this kit happening.
There are three main concepts that we hope your students will be able to understand after completing this kit. First of all, we want them to understand the process of diffusion, which is a passive process requiring no energy, in which the particles randomly move from an area of high concentration to an area of low concentration. Second, we're going to look at equilibrium across a membrane, in which the number of molecules becomes even on both sides and there's no longer any concentration gradient. And third, we're going to look at selective permeability, which is a process across a membrane where salt concentrations may build up on one side, causing the influx of water from the other side in order to cancel out the concentration gradient.
So we thought a good way to start this lesson would be to put some questions up on the board, which we have some demonstrations listed on the second page of your handout. And these questions are meant to probe the students' current knowledge on the concepts in this kit, diffusion, equilibrium, and membrane permeability. And we'd like you to collect some answers from the students. They can be wrong, they can be right, it really doesn't matter, and just put them up on the board. The point is to get the students thinking right off the bat. And you can maybe spend 5 or 10 minutes doing this exercise, then leave all those questions and the answers up on the board. And then at the end of the lesson, we thought it would be helpful to revisit those questions and see if the students can pick out the answers that they had listed that were right, or if they have additional answers that they think would be appropriate now based on the knowledge that they've gained from going through the kit.
There are multiple materials required for these experiments. All should be included in your kit, and we will go through these in detail as we go through each individual experiment. Some additional safety issues. First of all, many of these experiments require hot steaming water. You can heat up water using the included heating block or using any nearby microwave. We ask that you will have the students come up one student at a time from each group and pour it into their cup so that there are not issues with handling the hot water. Additionally, for one experiment, students will need to walk around the room and may have a chance of banging into each other, so please choose responsible students so that there are not issues with this. And finally, there is experiments that will involve edible gummy bears, but for the use of the experiment, we hope you will instruct your students not to eat them. And students should wear gloves for all experiments because many of these materials can be hazardous.
So in this first section of the kit, we'll be going through the concept of diffusion. We have a pretty simple experiment set up that demonstrates the concept of diffusion using simply just some hot and cold water and some food coloring. So we'll go ahead and get it set up now and go through it for you. So for this experiment on diffusion, you'll need 2 cups, one which will have cooler tap water in it, and one which the students will come up to you and you'll pour in some steaming hot water into it. It's important to make sure that the level of the water is similar in both the cups, so there'll be a line on the cups to which you should pour the water up to. And then we'll be taking some basic food coloring and then using eyedroppers to drop 2 drops of food coloring into each of the cups at the same time. Now, when the students do this, they should also have a timer, and it'll be important to start the timer when they drop the food coloring into the cups. So we'll go ahead and do that. So 2 drops in each cup. 1, 2, start the timer. And then the point of this starting the timer is because we want the students to take note on whether or whether it's faster for the food coloring to spread out in the warm water versus the cool water. Now in the background material, we mentioned that diffusion takes no energy to take place. And this is true. However, molecules move faster if they're warm. Therefore, the warm water, the food coloring should spread out much quicker than in the cool water. And then to wrap up this experiment, you can poll the students at the end, asking them for which groups the food coloring moved faster in the hot water, which groups the food coloring they thought moved faster in the cold water, and which groups thought that it was basically equal between the two.
As you can see, in the warm water, the food coloring is completely dissolved equally throughout the water, while in the cool tap water, the food coloring is not equally distributed yet. So by our recording, it only took about 15 seconds in the hot water and is taking over 1 minute in the cold water. Therefore, in hot water, diffusion occurs faster, we can conclude.
So in this part of the teaching kit, we want to demonstrate the concept of equilibrium to the students. For this, we'll be using your actual classroom, and you'll either set up the classroom such that you have physical barriers such as desks or tables down the middle, or a line of tape. We'll provide some tape in the kit that you can use. And this line of tape or line of barriers will represent a cell membrane. And then the students are going to serve as molecules that can either be inside or outside of the cell. And the point of this exercise is to demonstrate that if molecules can move across a membrane, they will eventually by the process of diffusion, want to reach a state of equilibrium, where there are equal molecules on one side of the membrane as on the other side of the membrane. For this part of the demo, you're going to have your students collect on one side of an empty room that's been divided through the middle with either chairs or tape, as we mentioned before. Have your students extend their arms horizontally, and then tell them that they're to walk around their side of the room, but they can't pass the barrier in the middle, which represents a membrane. So walk around, please. And they're supposed to walk around and try not to hit anyone with their arms. You can do this for pretty much as long as you see fit. 30 seconds is probably a good amount of time. And then after an appropriate amount of time, tell the students to stop. And they can put their arms down. And now you can ask them whether it was easy or hard, or if they could move around quickly with so many students packed together and their arms extended. So Kevin, could you move around? Did you feel like you could move around as fast as you could with your arms extended?
No, it was hard.
As it should be. And now for the second part of the demo, this time the students are going to be representing molecules that can cross across a membrane. So the membrane, or the chairs or tape or whatever is dividing your room, is considered a semi-permeable membrane in regards to the students now. So again, have all your students, or not all, but the amount of students that you selected on the same side of the room. It's important for this part that there be an even number of students. And again, tell them to extend their arms. And they're allowed to walk around, but this time they're allowed to cross across the semi-permeable membrane in the middle, and they can cross back and forth as many times as they want. But again, they want to move around as fast as they can and not run into anyone. The idea we're trying to establish here is that molecules will want to even out on either side of a membrane if they're allowed to cross the membrane. So when you see that the students have approximately reached an equilibrium, or an even number of students on either side of the membrane, tell them to stop. All right, stop. And have them put their arms down and look around and take note and see how many students are on either side of the room. You can ask them, Chris, how many students are on either side of the room? Is it even?
It's even.
Look at that, it's even. And this is the concept that happens in your cells when molecules can cross across a membrane. They're going to want to cross across the membrane until they reach equal numbers on either side. And this is considered an equilibrium. So first off, for this part of the experiment, it's important to make sure the students are all wearing gloves so they don't get any iodine on their hands. To start, we'd like you as the teacher to gather the students around and demonstrate to them, like we said, that when you mix iodine and starch together, it creates a purple color so that they know what color they're looking for in their experiment with the Ziploc bag. So to do this, we have an aliquot of starch that you can just pour into a cup of water. The temperature of this water doesn't matter, it can just be normal tap water. And then be sure to stir that up. The starch won't dissolve, it'll just make a suspension so it'll stay cloudy. And then we have some iodine provided, and you can just pour that in to the starch and let it mix around. And after it mixes completely with the starch, you can see that it produces this purple color, which is quite dark here. The intensity of the purple will of course depend on how much starch and iodine is in there, but this should be a good demonstration to the students that once iodine and starch can touch and are no longer physically kept from each other, they create a purple color.
So now we will be doing part of the kit studying selective permeability and tonicity. Cell membranes are selectively permeable to molecules, meaning that certain molecules are able to pass across, while other molecules cannot. We will be demonstrating this using a bag of iodine put into a cup of starch water. Starch is a large molecule that is unable to pass across the bag, which represents a cell membrane. However, iodine is a small ion and can easily pass across. When starch and iodine react, they produce a purple color. And by observing where we can see this purple color, we can determine the presence of molecules passing across the bag, and therefore determine whether these molecules are permeable or not.
So then for this part of the experiment, first have one student from each group come up to you and place some steaming hot water in their cup. You can fill the cup up to the marked line that we'll have. Then have the students carefully carry that back to their workstation. And they each should have a tube of starch. Have them pour that tube of starch into their provided hot water cup. And then mix it up so that the solution turns cloudy, much like you did in the demo for them just a few minutes prior. And then once that is done, they need to make their semi-permeable bag of iodine. So they can take the provided Ziploc bag, and then we already have some iodine poured out for them in a tube. Just have them pour that into their Ziploc bag and close up the bag. It's very important that they're certain to get the bag completely closed. And then once they have the bag completely closed, they can dip it into the hot water and make sure the whole iodine-filled portion of the bag gets dipped in there. And then eventually the iodine will start moving out of the bag. This will take a little bit of time. It doesn't happen instantaneously. And while the students are waiting for this to happen, they can periodically kind of jostle the cup a little bit. And while they're waiting on that to happen, we provided a few questions within the handout. One of them actually addresses the fact that this takes a little bit of time and what some of the factors that they think might be that makes it go slower or faster for different groups. So while they're waiting for this iodine to leave the bag and interact with the starch, you could have either the groups discuss amongst themselves the answers to these questions and then maybe poll the whole class and ask them for some of their answers. Or you could have a whole class discussion on what they think might be happening right now. It's important to not give it away that that iodine is moving out of the bag and reacting with the starch. We want them to come to that realization on their own based on where they see a purple color develop. So as you can see here, we actually have some of the iodine starting to come out of the bag. This is at the very beginning of the process, and it's just turning these starch particles that it's touching purple right here, not the whole bag quite yet. But this is how it'll start. And when this sort of thing starts to happen, you can have the students tap their cup or shake it a little bit to try to speed up the process. But it'll still take some time for the entire cup of water to turn purple. It is kind of cool to just watch it slowly trickle out and turn that starch purple in the very beginning. So as you can see here, our starch solution is getting much more purple. The iodine and starch has been sitting together for about 15 minutes. If it takes longer than this during your class period, and if you're running out of time, feel free to have the students set this part up, and if they're not seeing a really vibrant purple color change, you can have them leave it overnight or until the next class period and then observe the final color at a later date. But hopefully you'll be able to see it within the class period. As you can tell here, it is turning quite purple and the iodine is coming out of the bag as we would expect it to. So as you can see here, like I said, the starch has turned purple, and if we take the bag out of the solution, you can see that there's no purple color inside of the bag, but there is purple color inside of the cup. And what the students are supposed to conclude from this is that iodine moved out of the cup and reacted with the starch on the outside of the bag, meaning that the plastic bag, which served as the cell membrane, was permeable to iodine but was not permeable to the starch molecules.
As a result of the selective permeability of cell membranes, salt may accumulate on one side of the membrane, either inside the cell or outside of the cell. In order to achieve equilibrium, this means that water must therefore move outside of the cell or into the cell in order to equilibrate the concentration of these salt molecules. To demonstrate this concept, we will be using gummy bears to represent a cell, and the sugars of the gummy bear will represent salt molecules within a cell. For this demonstration, we will be using gummy bears as examples of cells, and we will place them into either a cup filled with water or an empty cup. Students will place one each of the gummy bears into the empty cup and into the cup of water. And this is tap water. These cups will then be left overnight until the next class period. When students return, they should empty the water out of the cup, of the water containing cup, and then compare the sizes of the gummy bears between the water cup and the empty cup to see whether the gummy bear has shrunk, grown, or stayed the same size. The reason why we are comparing the different sizes of these gummy bears is because selective permeability can result in a phenomenon known as tonicity, where water will move quickly into or out of a cell in order to equilibrate concentration of the salts. In this situation, the water represents what's known as a hypotonic solution because it contains much less salt than the inside of the gummy bear. In this situation, water should move quickly into the gummy bear in order to equilibrate the salt concentration, causing it to swell, similar to what happens in actual cells. The alternate situation involves what's called a hypertonic solution, where the surrounding solution outside a cell contains more salt than the inside of the cell. This forces water to leave the cell, and the cell shrinks. I have now let the gummy bears sit in the water overnight, and as you can see, I have removed the water from the cup containing the water, and there is a marked difference in the size of the gummy bears. It is a little less obvious on camera, but the gummy bear that was in the water is much larger, is also much more fragile and bloated. You can see part of it is actually broken off. Now, we did not include the opposite experiment in this demo, in which we put the gummy bears in salt water, because since the inside of the gummy bears is so sugary, we found that it was very difficult to get water salty enough to make it hypertonic. However, if you choose, you may perform this experiment with your students anyway, and then explain possibilities why it may not work.
To account for this, we have simply included a question asking the students to guess what would happen if the gummy bears were placed in extremely salty water.
Thank you so much for using our kit. When you're done with all the experiments, we have some questions at the end of the handout. These happen to be the same questions that you went over with the class in the beginning of the lesson. It'd be nice to address these questions again and see if the students have any more or can pick out the correct answers to all the questions based on the concepts that they've learned throughout the demos. Additionally, we have listed questions throughout the handout that the students can do while the experiments are happening and also after each one of the experimental sections to really help solidify the concepts that we're trying to get across with this teaching kit.
Additionally, we have included a fill-in-the-blank exercise as a final evaluation of these students' understanding of the concepts presented in this kit. We hope that that will be a good example of testing whether they have really, this kit has helped them to further understand the concepts presented. We hope that you and your students will fill out the included evaluations in order to help us better improve this kit for the future. And if you have any questions, please contact Jen Mosher at the included addresses. Thank you.
And happy experimenting!